Hello all and welcome back. So we will be proceeding ahead with the different procedures for solving the ABO group discrepancies. So we will be discussing about the enzyme treatment in which the weaker antigens are enhanced and it helps in detection of the weaker subgroups. We will also be discussing about the adsorption and elution methods which are used for detection of the weak antigens. So both of the procedures are a bit lengthy and not very commonly followed in all the institutes and facilities. So it might feel a little bit overwhelming, but you can also take a guide from the ABB technical manual. You can keep the methods with you and then follow the video, and that will give you a better insight as to how the techniques are to be done. After the techniques are described, we will discuss the entire summary and we'll have a brief question and answer session related to the topic. So we have Dr. Soumya again discussing the various ways in which we can resolve group discrepancy. So we'll be discussing a case-based scenario in which we'll discuss the enzyme method as well as the adsorption and elution method. Thank you.
A very good morning everyone. So today we are going to discuss about a very interesting blood group discrepancy of a patient which was received to our hospital for pre-transfusion ABO grouping. The patient was a 47-year-old female with diagnosis of left ovarian cystic lesion who was posted for cystectomy and required 1 unit of PRC for surgery. The patient was para 3 live 3 and all were normal deliveries. There was no surgical, transfusion or significant drug history. On initial forward and reverse grouping, the results of forward group are: there was no reaction in anti-A, anti-B and anti-AB and strong 4+ agglutination on anti-D, which was suggestive of O positive. On reverse grouping, there was strong 4+ agglutination on A1 cell and no reaction in B cell, O cell and autocontrol, which was suggesting towards B group. So here the forward and reverse grouping are not matching to each other. Hence there is a discrepancy in this case. Further on, we obtain fresh sample from the patient. With fresh sample, the red cells were thoroughly washed with saline at least 2 to 3 times. This washing will help us to eliminate the reaction associated with plasma proteins or autoantibodies. So with fresh sample, we further on proceeded with forward and reverse grouping at all 3 temperatures, that is room temperature 37 degrees Celsius and 4 degrees Celsius. On forward grouping, there was no reaction in anti-A, anti-B and anti-AB on all 3 temperatures and strong 4+ agglutination in anti-D on all 3 temperatures, which was again suggesting towards O positive. On reverse grouping, there was strong 4+ agglutination on A1 cell at all 3 temperatures and no reaction in B cell, O cell and autocontrol, which was again suggesting towards B group. So here the discrepancy is not resolved even after performing at all 3 temperatures. So when we thought that what were we missing, we found that there was strong 4+ agglutination reaction on A1 cell at all 3 temperatures, which was suggesting that there is a strong antibody and there can be missing antigens in this case. To detect the missing antigen in this case, we further on proceeded with enzyme treatment. Enzymes like papain and phesin can be used. These enzymes will modify the RBC membrane and causes enhanced antibody binding to the RBCs, thus helping us to detect the missing antigen. Coming to the requirements which are needed. First, monoclonal anti-A, monoclonal anti-B and anti-AB. The enzyme which we are going to use is phesin solution and a phesin control to check if the enzymes are adequately treated with the red cells. Coming to the samples which are required, patient red cells and serum is separated and kept in 2 separate clean label test tube. With the patient red cells, 3% red cell suspension is made and kept in separate clean label test tube, and in the other tube we have taken 3% washed untreated O red cells, which acts as a control in this case. The enzyme which we are going to use here is the Fissin solution. According to our manufacture instruction, Fissin comes in concentrated form. Hence, it is to be diluted in the ratio of 1:9. So, in a clean test tube labeled as diluted Fissin, I am going to now add 2 drops of Fissin solution and 18 drops of saline to it. So in the clean label test tube, now first I have added 2 drops of Fissin and now I am adding 18 drops of saline into it. So now our diluted Fissin is now ready to use. Next, take 2 separate clean test tube and label as enzyme treatment of patient and enzyme treatment of control to perform the test. Now we are going to treat the red cells with enzyme according to our manufacture instruction. So for that we are going to add equal amount of diluted Fissin and 3% red cells. So now I am taking 500 microliter of diluted Fissin and adding in both the test tubes. First I am adding in enzyme treatment of patient. Next same 500 microliter I am taking and add in the enzyme treatment of control O cells. Now we are going to add red cells into the diluted Fissin. First I am going to add 500 microliter of patient red cells into diluted Fissin. So 500 microliter of red cells is being taken now and add into the respective labeled test tube, that is enzyme treatment of patients. So this tube contains 500 microliter of Fissin and 500 microliter of patient red cells. Next, after changing the tips of the micropipette, 500 microliter of control, that is O red cells, is being taken now. And it is being added into the respective labeled test tube, that is enzyme treatment of control O cells. So this tube now contains 500 microliter of Fissin and 500 microliter of control O red cells. Now we have to incubate both the test tubes at 37 degrees Celsius for 10 to 15 minutes. After incubation, wash both the red cells with saline at least 3 times. So now we have taken enzyme treatment of patient tube and filled 3/4 with saline. And then in the enzyme treatment of control, fill the test tube with 3/4 with saline. And then centrifuge the tube by properly balancing according to your institutional SOP. After centrifugation, remove the supernatant saline. The washing is to be performed at least 3 times. This is our third wash and we have removed supernatant saline. Now we are removing the supernatant saline in the enzyme treatment of control tube. And these red cells should be made 3% red cell suspension to perform the grouping. Now enzyme treated red cells are ready to use. So to treat the red cells, first we diluted Fissin, then equal amount of Fissin and red cells were added and then incubated and washed with isotonic saline and made as 3% red cells to perform grouping. Before performing grouping, we need to know if the cells are adequately treated with enzyme. So for that we have Fissin control solution. Here, according to our manufacture instruction, twice the amount of red cell Fissin control is to be added in enzyme control tube. Hence, 4 drops of Fissin control is added in enzyme control tube containing the cells. Next, tube is to be centrifuged with proper balancing according to the manufacturer instruction. For the cells to be adequately treated with enzyme, control should give 3 plus to 4 plus agglutination reaction. After centrifugation, the tube is now taken out. Gently suspend the button of the tube and look for agglutination reaction. Here we have got strong 4 plus agglutination reaction, which shows that cells are adequately treated with enzyme and hence the patient red cells are ready to use now. Now with enzyme treated red cells, we are going to perform forward grouping as we suspected a missing antigen in this case. I have taken a clean test tube and labeled as enzyme anti-A patient, and in that tube add 1 drop of anti-A. Next, in separate clean test tube, label as enzyme anti-A control. To this tube add 1 drop of anti-A. Take another clean test tube and label as enzyme anti-B patient. To this tube add 1 drop of anti-B. Take another clean test tube and label as enzyme anti-B control. To this tube add 1 drop of anti-B. Take another clean test tube and label as enzyme anti-AB patient. To this tube add 1 drop of anti-AB. Take another clean test tube and label as enzyme anti-AB control. To this tube add 1 drop of anti-AB. Now to each respective tubes we are going to add red cells. First, the enzyme treated patient red cells, we are going to add 1 drop in anti-A patients, anti-B patients and anti-AB patient tubes. Enzyme treated O cells, we are going to add 1 drop in anti-A control tube and then anti-B control tube and last anti-AB control tube. Incubate for 30 minutes at room temperature all the test tubes. After 30 minutes of incubation, the tubes are kept in the centrifuge, equally balanced and centrifuged according to our manufacturer instructions. After centrifugation, gently suspend the button and look for any agglutination. First, we are checking in control tubes. As we can see in the picture and video, there is no agglutination at all in either of the tubes, anti-A, anti-B and anti-AB. In all the 3 control tubes, which suggests that cells are adequately treated and tested properly. Next, coming to the patient sample, there is no reaction in anti-A, anti-B and anti-AB, which again suggests that it is O group. Hence, now also the discrepancy is not resolved even after enzyme treatment. Even after by enzyme treatment, the group discrepancy is not resolved. Hence, some weak ABO subgroups are too weak to be detected by direct agglutination even after cold temperature and antibody enhancement. The presence of A antigen, B antigens or both requires adsorbing anti-A or anti-B to red cells followed by elution of bound antibody. The elevate is then evaluated for the presence of anti-A and anti-B by testing against A1 or B reagent red cells. The requirements which are required are: first, the patient red cell which is to be tested. Next, we have taken here human anti-B because we are suspecting weak B antigen, hence we have taken anti-B. Monoclonal ABO typing reagents are not used because it has sensitive to changes in pH and osmolarity. Hence, the reagents may not be suitable for use in adsorption and elution studies. Next, we have taken 3 examples of group O red cells and named as O1, O2 and O3, and 3 examples of B red cells, that is B labeled as B1, B2 and B3. Coming to cold adsorption technique, the first step is wash 1 ml of patient red cells with saline at least 3 times. So now we are adding patient red cells with 3/4 of the saline. After this, the test tube is kept in centrifuge and properly balanced. Centrifugation is to be done according to manufacturer instruction. After centrifugation, the supernatant is to be removed and the washing is to be done at least 3 times. After the last wash, the supernatant is to be discarded. Now add 1 ml of human anti-B into the patient red cells. As we suspected a weak B variant, we have taken anti-B reagent. So we have taken 1 ml of anti-B and it is being added into the patient washed red cells. After adding, mix red cells with the antibody. Now keep the red cell mixed with antibody at 4 degrees Celsius for 1 hour. During 1 hour incubation, mix the tube occasionally. After 1 hour of incubation at 4 degrees Celsius, we are taking the test tube and centrifuge the mixture to pack the red cells. Now we are placing the tube into the centrifuge and with proper balancing, centrifuge the tube according to the manufacturer instruction. So after centrifugation, we can see here the red cells have settled down and the reagent is present at the supernatant. So now remove all the supernatant reagent from the red cells. After removing all the supernatant from the red cells, now transfer the patient red cells into a clean labeled test tube. So now we are transferring the patient red cells into clean labeled test tube, which we have labeled as patient red cells. Now we are going to wash the red cells with saline stored at 4 degrees Celsius for at least 8 times. So for washing, fill the red cell tube 3/4 with 4 degrees Celsius saline. Centrifuge the tube with proper balancing. And then after centrifugation, remove the supernatant saline. The same procedure is to be repeated at least 8 times. So again fill the red cell tube with 3/4 of saline stored at 4 degrees Celsius. Centrifuge the tube and at the final wash, save the supernatant fluid to test it parallel with the eluvate. So this is the final wash. So now I am going to preserve the final supernatant. I have taken clean labeled test tube and labeled as final supernatant. Now we are going to transfer the final supernatant into the clean labeled test tube. So our final supernatant we have preserved. We are going to perform heat elution. Heat elution will elevate the ABO antibodies, which are mostly IgM in nature, from the red cells. The mechanism is that with increase in temperature, it will dissociate antibodies from the red cells. The requirements which are needed are patient packed cells, which we obtain from cold adsorption technique, and 6% albumin. The first step is add equal amount of 6% albumin into washed packed cells. So now we have taken 1 ml of 6% albumin and now we are adding into 1 ml of patient packed cells. Now cover it with the parafilm and mix it gently. Keep the tube at 56 degrees Celsius in the water bath for 10 minutes. During this time, agitate the tube periodically. After 10 minutes of incubation at 56 degrees Celsius, centrifuge the tube at 900 to 1000 gram for 2 to 3 minutes by properly balancing the centrifuge. After centrifugation, take out the tube. We can see in the supernatant we have got the elevate. So this is after centrifugation. We can see a light pinkish red supernatant which is present. So we have taken a clean label test tube and now we are going to transfer the elevate into the test tube labeled as elevate. Take out the elevate very gently without shaking the patient red cells. Supernatant should be removed immediately as soon as possible after centrifugation. Now we have got the elevate in the test tube and this should be tested in parallel with the supernatant saline. We are going to test it with the elevate. Take 3 clean label test tube which is labeled as B1, B2 and B3 and another 3 clean test tube which is labeled as O1, O2 and O3. And 3 examples of group O red cells and 3 examples of group B red cells. Now add 1 drop of B1 red cells into B1 label test tube, B2 red cells into B2 label test tube, 1 drop of B3 red cells into B3 label test tube and 1 drop of O1 red cells into O1 label test tube, 1 drop of O2 red cells in O2 label test tube and finally 1 drop of O3 red cells in O3 label test tube. Next we are going to add 2 drops of elevate into all 6 tubes. Now we have added in B1. Next in 2 drops into B2 test tube. Now we are adding in B3 test tube. Now in O1 test tube. Next in 2 drops of elevate in O2 test tube. Finally 2 drops of elevate in O3 test tube. Ensure that you use separate pipettes for taking out the reagents and red cells. Next, keep the test tube for centrifugation by properly balancing. So we have 6 test tubes. Properly balance the test tube and keep in centrifugation and centrifuge according to manufacturer instruction. After centrifugation, gently suspend the button and look for agglutination reaction. So first in O1, O2 and O3 cells, we have got no reaction. Coming to the results of elevate in B1, B2 and B3 cell, here we can see there is 2 plus 2, 3 plus agglutination reaction in the test tubes, which shows that weak B antigens are identified. Hence the patient blood group is subgroup of B group. Test the elevate and final wash in parallel against 3 examples of group O red cells and 3 examples of group B red cells. So here we are now going to add 1 drop of red cells into all the test tubes. So B1 red cells we are adding now in clean label test tube, which is labeled as B1. Now B2 red cells 1 drop in B2 clean label test tube. Next, 1 drop of B3 in another clean labeled test tube, which is labeled as B3. Next, 1 drop of O1 cells into O1 test tube, 1 drop of O2 cells, and then 1 drop of O3 cells into clean label test tube. We have the final supernatant, which we have preserved after the final wash. Now we are going to add 2 drops of final supernatant into all 6 tubes. Now we have added in B1 and B2 cells. Next B3 test tube. Next O1 test tube. Next to the O2 test tube. Finally 2 drops into the O3 test tube. Centrifuge the tube and look for agglutination reaction. So there are 6 tubes. So by properly balancing, centrifuge the tube according to manufacturer instruction. After centrifugation, gently suspend the button and look for agglutination reaction. So we can see there is no agglutination on O1, O2 and O3 cells. Next, in B1, B2 and B3 cells, we can observe there is no agglutination reaction. So the final wash is non-reactive with all the cells tested, which shows that our elevate results are valid. If the wash solution is reactive with antigen positive red cells, the elevate results are invalid. The reaction can occur if unbound reagent antibody was not adequately removed by washing before the elution step or by dissociation of bound antibody during the wash process. So here our elevate results will be valid because our final wash is non-reactive with all the 6 test tubes.
So coming to an overview of the entire procedure which was discussed, we'll have a brief summary of the entire thing. So as we received the sample of the patient, we saw that the forward grouping showed a group of O group and RHD positive, whereas in the reverse we got a reaction with A1 cell suggesting a group of B. So the forward was O and the reverse was B, which led to a group discrepancy. Now the patient sample were requested again, a fresh sample was obtained, a proper washing was performed and the test, the forward and the reverse grouping were done at various temperatures, but the test results did not vary and it remained the same. So the forward still showed O positive and the reverse showed a B grouping. So proceeding ahead to solve the discrepancy, we went through the enzyme treatment. We treated the cells with ficin, which is an enzyme, and then we got the results with the enzyme treatment. Here we were not able to resolve the test results. The forward grouping still yielded O positive at all 3 temperatures and it showed B group in the reverse grouping as well. Many times, in many cases, the enzyme treatment itself is sufficient to resolve the case and the antigens and the groups are resolved, the discrepancy is resolved. But in this case, we were not able to resolve this. So we proceeded further ahead for the adsorption and elution. So after the cold adsorption and heat elution, the results of eluate and supernatant were tested with 3 samples of group O red cells and 3 samples of group B red cells. So the results of eluate showed no reactions with the O cells, the O1, O2 or the O3 cells, whereas it showed a 3 plus and 2 plus reactions with the B group of cells, which shows that the test was valid and the group was a weaker subgroup of B. And the last supernatant was also tested against the O red cells as well as the B red cells, and we did not notice any reaction, which again revalidates the entire test process. So thank you, Soumya. So with that, we have come to an end of the procedures to solve the ABO group discrepancies using the enzyme and the cold adsorption and heat elution techniques. But there are a few questions which are often asked, and also we have queries while we perform these tests. So I would like to discuss them with you. So my first question to you is, what are the enzymes? I mean, how do they act on the red cell antigens, or how do they affect the expression of the red cell antigens? If you can elaborate on it.
Yes, ma'am. Enzymes work by modifying the RBC membrane by exposing the hidden antigenic sites, reducing steric hindrance, and by reducing zeta potential. This helps to increase the antibody uptake and thereby increasing the antibody detection sensitivity. The red cell antigens which are increased by enzyme treatment are ABO, RH, KIT, Lewis, P and I systems. And also enzymes can abolish the reactions by cleaving or altering the antigen itself. So the antigens which are decreased by enzyme treatment are Duffy and MNS systems. And also red cell antigen which is unaffected by enzyme treatment is the KEL system.
So yeah, okay. So it has a different effect on different antigens. In few of the antigens, like you said, the RH, the ABO system, they get enhanced by the enzyme treatment, and the enzyme systems like the MNS and Duffy, they get suppressed. So apart from fissin, which was used in the current example, can you name a few other enzymes which are used in the immunohematology investigations?
Thank you for the question, ma'am. Other than fissin, the enzymes which are widely used in blood bank are peptide and trypsin. These are proteolytic enzymes that cleave the red cell antigens on the RBC membrane.
So right. So during the enzyme treatment, I noticed that you have used a group O red cells, wherein they acted as a control and you have treated them with enzymes. So what if these group O enzyme treated red cells give a positive reaction when they are tested against the anti-A, anti-B or anti-AB reagents? So what is the inference when this happens?
Yes, ma'am. If there is reactivity in the enzyme treated control O red cell, it indicates that there is excess enzyme treatment. No ABO interpretation can be made if there is reactivity in the control enzyme treated red cells.
Thank you for that. Now, moving on to the next topic or the next procedure which you showed was the adsorption and elution technique. So, the human anti-B antisera was used for the test instead of the monoclonal commercially available antiseras. So, could you explain what was the reason behind this? Can we go ahead and use the monoclonal reagents which are easily available?
Yes, ma'am. In adsorption and elution technique, human anti-A or anti-B are used because monoclonal antibodies are so specific and may not contain certain specificities to sensitize some of the weaker antigens. And also, monoclonal ABO typing reagents are sensitive to pH and osmolarity and hence may not be used in adsorption and elution test.
So at the end, you have washed these cells around 7 to 8 times, and the last wash the supernatant of the last wash was retained and then tested against the antigen positive cells. So can you explain the significance of performing this step, and what if the supernatant tests positive with the antigen positive cells?
Yes, ma'am. If the wash solution is reactive with antigen positive red cells, the elevate results are considered invalid. The reaction can occur if there is unbound reagent antibody which was not removed properly by washing before the elution process, or by dissociation of bound antibody during the washing process.
So thank you so much, Soumya, for answering all our questions and resolving our queries. If you face any other queries, please drop them in your comment section. We would also like to have a feedback of yours if you have come across any interesting case during solving the ABO discrepancies, or any other queries or problems you face during the procedures, and we would be happy to help. We will proceed with the part 3 and part 4 in the upcoming episodes. So please do subscribe not to miss them, and stay tuned. Thank you.